So as a population ecologist, we've got a huge environment to attempt to survey. We've got a large number of plants, we've got a large number of animals, and in order to capture a true representation of how they are growing and where they are growing, we need to consider an experiment that is valid, accurate, and reliable. In order to do this, we could attempt to simply walk through this environment and count every single plant and every single animal. But inherently, that has a huge range of problems. It would be unlikely that we'd be able to achieve an accurate count. We'd miss animals, we'd miss plants. We'd be unable to do a reliable one because simply trying to replicate that again in the future would be met with future issues of trying to find the same plants and the same animals once again. Finally, it wouldn't be valid. If you can't compare experiments that have been conducted on separate occasions, it's simply not a valid experiment. To ensure our population survey is accurate, reliable, and valid, instead of exploring this whole ecosystem, we're going to divide it into smaller pieces. The item that we're going to use to do this is called a quadrat. Today, it's a square, 1 metre by 1 metre, and this quadrat is going to be used to divide up the whole ecosystem into much smaller components. Those components can then be counted accurately, repeated for reliability, and because we can then compare the results, it is now valid. Today, the metre by metre quadrat is chosen because the smallest animals, the bembexium, the small crabs, can be identified in this size, but also our larger plants can also be captured. If we were studying larger animals such as rhinos or elephants, obviously our quadrat would need to be much larger, and vice versa, small organisms such as microscopic bacteria or viruses might need an incredibly small quadrat in order to support ecologists studying those systems.